Lesson 3, Chapter Photosynthesis in Higher Plants. Students, in the last video, I discussed about the light reaction that takes place in the thylakoid of the chloroplast in the green parts of the plant body. That is generally the young stem and the green leaves. And the light reaction, it was all about generating the assimilatory power in the form of ATP and NADPH, which is produced during the second phase of the light reaction that takes place within the thylakoid in the chloroplast. And this it happens with some electron acceptors, and this whole process it was called as electron transport chain, that we also call it to be photophosphorylation. And in that photophosphorylation we studied in two types. One is cyclic photophosphorylation and the other one is non-cyclic photophosphorylation. These two electron transport chains, that is cyclic and non-cyclic, that took place within some photosynthesis which are made up of chlorophyll pigments, a special type of chlorophyll pigments that can receive the photon particles of wavelength of around 700 nanometer, which we call it to be PS1, and some they could receive the wavelength of light in 680 nanometer, so that we call it to be PS2, that is photosystem 2. So in today's video I'm going to discuss a small hypothesis. This is called as chemiosmotic hypothesis, in order to explain the photophosphorylation, or that how this assimilatory power that is formed there in the thylakoid membrane, or what happens during that whole process in the thylakoid as well as there in the stroma of the chloroplast. So generally you know that a photophosphorylation, or that is the process of phosphate group transfer into ADP to synthesize the energy rich ATP molecule, making use of light as an external energy. So as you know that in order to do any work we always require the ATP molecules. So the production of the carbohydrate, or the synthesis of the carbohydrate there in the chloroplast, it also requires energy. And in that those electron acceptors such as ferredoxin, plastocyanin, plastoquinone, they had undergone some kind of electron transport chain, and in that way they produce two very important molecules. One is ATP and the another one that is NADPH2, and these two are both energy rich molecule. Now how this ATP is produced? So there is a mechanism of ATP synthesis in photosynthesis that takes place in the chloroplast, has been explained by Mitchell in the year 1961, and this is called as chemiosmotic hypothesis. Now what happens in this chemiosmotic hypothesis? So according to this hypothesis, the ATP synthesis is linked to development of a proton gradient across the thylakoid membrane. So as you know that the ATP synthesis that takes place there inside the membrane of the thylakoid particle that is present inside the chloroplast, and in that you also have known that during this whole process water gets split, and that splitting of water that is called as photolysis of water, and in that particular process there one proton molecule was formed. That is, you can refer to this reaction that H2O, when it gets broken down then we get one hydroxide ion and one hydrogen ion, which is the proton here. And in that, in order to produce two oxygen molecule, which happens during the photolysis of water, so there we find that some extra protons they get started to increase there in the thylakoid membrane. So therefore it is said that according to this hypothesis, the ATP synthesis, it is linked to, so that is very much dependent upon the proton gradient, that is increase in the number of the H plus ion across the thylakoid membrane. And the actual site of this ATP synthesis is the ATPase complex. The ATPase complex, there is the enzyme where this ATPase, that is ATP synthase enzyme, is present there, and this is generally present there in the non-appressed portion of the thylakoid membrane, particularly towards the margin. And during photosynthetic electron transport, these hydrogen protons, they accumulate in the thylakoid space, and they started to increase in their concentration. So these hydrogen proton, they are released into thylakoid space after each split of the water molecule during this photo-oxidation process, and in that way this there occurs a difference in the proton molecules inside the thylakoid membrane and outside the thylakoid membrane. So outside the thylakoid membrane, so that is the stroma. So stroma, they also bears an amount of the hydrogen ion, and inside the thylakoid, the hydrogen ion concentration that gets increasing because of the photolysis of water that takes place during the presence of light. And in that way, there is a difference in the both the sides of the thylakoid. So an increase in the number of this hydrogen proton in the inner space of the thylakoid causes the pH of one side of the membrane to decrease. So at the same time, the pH of the thylakoid membrane towards stroma, that starts increasing due to the utilization of this proton or H plus ion during the conversion of the plastoquinone that is present inside the thylakoid, and this plastoquinone that is converted to plastoquinone hydrogen molecule, and at the time of reduction of this NADP plus molecule. So this difference in the hydrogen concentration of the two sides of the thylakoid membrane leads to building up a transmembrane proton concentration gradient. That means the inner side, there the proton, that is H plus ion, so that is increased there, and outside of the thylakoid, that is towards the stroma, as it starts decreasing. So in this way, an electrochemical proton gradient of, or potential gradient of these proton molecules, that starts to create a driving force behind the conversion of the ADP and phosphate molecule to produce the ATP molecule. And this whole process that gets happen there inside the wall of the thylakoid and towards the outer part of the thylakoid. So this, it was explained with the help of this chemiosmotic hypothesis. So this chemiosmotic hypothesis, it is nothing, it is just the development of the proton gradient that takes place inside the thylakoid, which makes the concentration of the H plus ion inside the thylakoid more, leading to lowering of the pH, and the proton gradient that takes place towards the proton molecule that is present outside the grana, or that is towards the stroma, that leads to the increase in the pH because there is less hydrogen compared to the inner part of the thylakoid. So this whole phenomenon that goes on inside and outside the thylakoid membrane, so that leads to the production of the ATP. So this is just a small hypothesis which was put forward by Mitchell in the year 1961. And in that way, we get the assimilatory power, that is ATP, which is used in the synthesis of carbohydrate in the dark reaction. So let's come to the dark reaction now. So as you know, what is dark reaction? So dark reaction, it is the second step of photosynthesis, where light is not an important factor. And this dark reaction, it takes place in the stroma of the chloroplast. This is the matrix of the chloroplast. The dark reaction, this is where the carbohydrate molecule, that is glucose, is produced. The dark reaction, it is mainly explained with the help of Calvin cycle. So let's discuss the Calvin cycle now. The dark reaction, which is also called as the biosynthetic phase, occurs in the stroma of the chloroplast, where the products of the light reaction, that is ATP, are used to incorporate carbon from the carbon dioxide to carbohydrate. As you know, that in the light reaction, there was no such occurrence of carbon products that we generally get in the chemical reaction of photosynthesis. So now in the dark reaction, usually the ATP will come to act in order to produce the carbohydrate in the form of glucose from carbon dioxide, which will be received from the air. And in that way, the dark reaction will be completed. So although the reaction, it is called to be dark reaction, but it does not, and it is said that it does not require light. That means that it is independent of light. But usually this process, it occurs in the light and continues for a very brief period after a plant is kept in dark, as long as NADP and ATP are available. So this particular process, it takes place both in dark or in light condition. So therefore I have said that it is independent of light. So a better understanding of this mechanism of the carbon dioxide fixation, it was studied by Melvin Calvin, Benson and their colleagues in California in the mid-50s. And in that, they identified an intermediate compound and a detailed pathway of carbon in photosynthesis called Calvin cycle is what we are going to study now. So Melvin Calvin, he had done a very important contribution in the study of this photosynthetic process. And therefore, for this particular pathway that they have created, so he was awarded Nobel Prize in the year 1961. So let's start with the reactions that happens in this Calvin cycle. Calvin cycle, this is also called as C3 cycle. Calvin cycle can be studied by splitting the complete cycle into three phases. And these three phases are carboxylation phase, reduction phase, and regeneration phase. So let's discuss the first step, that is carboxylation. So it is the initial reaction of Calvin cycle in which the atmospheric carbon dioxide combines with ribulose 1,5-bisphosphate. I'm repeating again, the carbon dioxide combines with ribulose 1,5-bisphosphate. That means this RUBP that we call in short, this is the carbon dioxide acceptor molecule there within the chloroplast in the stroma. So this RUBP molecule that is present in the stroma and that receives the carbon dioxide molecule. And it forms an unstable compound which splits into two molecules. One is 3-phosphoglyceric acid. Two molecules of 3-phosphoglyceric acid. So this is a very unstable compound, that is 3-phosphoglyceric acid. And this 3-phosphoglyceric acid, so this is a 3-carbon compound. And in this whole process, an enzyme, a very important enzyme, that is ribulose bisphosphate carboxylase oxygenase, that is in short called as Rubisco, which is the most abundant protein on earth, and it is present 16% there in the chloroplast protein. So that helps in producing this 3-phosphoglyceric acid during this carboxylation process. That means when carbon dioxide that is accepted by this RUBP molecule, which is ribulose 1,5-bisphosphate, that is catalyzed by the enzyme Rubisco, and in that way it produces finally a stable compound which is called as 3-phosphoglyceric acid, or that is also called as 3-phosphoglyceraldehyde. So this is how carboxylation process takes place. That is the carbon dioxide fixation there in the stroma of the chloroplast. Now as this 3-phosphoglyceric acid that is produced, the next step it begins. So that is reduction phase. So this 3-phosphoglyceric acid, so that is reduced to dihydroxyacetone, and from this dihydroxyacetone it goes into the glycolytic pathway, but in a reverse way. Dihydroxyacetone that forms fructose 1,6-diphosphate. From fructose 1,6-diphosphate it forms fructose 6-phosphate. From fructose 6-phosphate it forms glucose 6-phosphate, and from glucose 6-phosphate it forms finally the glucose molecule. And as this is the reverse of glycolysis, so this second step, which is the reduction step phase, that is also called as reverse glycolysis. And finally, from this 3-phosphoglyceric acid, which also forms the hydroxyacetone, and from that it takes this glycolytic pathway where it forms at first the fructose 1,6-diphosphate, then fructose 1,6-diphosphate to fructose 6-phosphate, then fructose 6-phosphate to glucose 6-phosphate, and from glucose 6-phosphate to glucose. Till this process, this is how we get the glucose molecule by the reduction process. And finally, again, as the RBP needs to be synthesized for accepting the next carbon dioxide molecule, so the last phase, that is the regeneration phase. Regeneration phase, that is of the ribulose 1,5-bisphosphate molecule. So this ribulose 1,5-bisphosphate, so that is regenerated from many pathways, such as one that is regenerated from 3-phosphoglyceric acid, where this 3-phosphoglyceric acid it forms ribose 5-phosphate. From ribose 5-phosphate it forms, which is a 5-carbon compound. Then from this it will produce ribulose 5-phosphate, and ribulose 5-phosphate, then from that by breaking down one ATP molecule it forms ribulose 1,5-bisphosphate. So in that way ribulose 1,5-bisphosphate that is regenerated, or the REBP molecule, which we call it in short, that is regenerated for accepting the next carbon dioxide molecule. Or otherwise it could also be regenerated from this dihydroxyacetone molecule also, which is also a 3-carbon compound. Now see, this whole process, this is called as carbon C3 or carbon tree, because its first stable product, that is a 3-carbon compound, that is 3-phosphoglyceric acid. So this Calvin cycle or Tracy cycle, C3 cycle, so that involves 3 steps, mainly 3 steps. One is carbon fixation step, which is also called as carboxylation phase, then reverse glycolysis process, that is reduction phase, and regeneration of RBP process. So that we call it to be regeneration phase. And this is how Calvin cycle or sweet recycle is finished, and we get the glucose molecule from carbon dioxide. Now let's summarize this Calvin cycle, which is a part of the dark reaction, and the light reaction in the chemical reaction that we get or that we study during the photosynthesis process. So the chemical reaction, that is carbon dioxide, water, that yields glucose, oxygen and water molecule. So this carbon dioxide molecule that comes in the next step, that is in the dark reaction, that gives us this glucose molecule and the water molecule. As in this whole reduction process, water molecule gets also splitted. So this water molecule that we get it in the dark reaction, and the water molecule that is present or that is involved in the process of photosynthesis, so that gives us oxygen molecule. So in order to produce one glucose molecule, it requires 6 carbon dioxide molecule, and in order 6 carbon dioxide molecule and this 6 oxygen molecule that is produced by splitting of 12 water molecule, and we get this 6 water molecule along with the glucose molecule from the whole process which is involved in the both light reaction and the dark reaction. So this is how we can get the glucose molecule, 6 molecules of oxygen and 6 molecules of water molecule in the whole photosynthetic process, which is a part of the dark reaction. So in the next class, I will discuss one more cycle which is also involved with this dark reaction, which is called as Hatch and Slag cycle.